Okay, thank you very much, and good morning. And I'm very grateful to Sandrine for inviting me here to this nice city. As I told him, I'm sorry to say, but I'm still first time in Austria, while we are already long or old members. We have been in European Union for a long time already, 10 years, but still there are places you haven't visited in Europe. I'm coming from Tartu, from Estonia, and the topic of my lecture is okay, sorry. It's about skin diseases. Maybe I can use mouse. I can use mouse here. Okay. Sorry, scroll. Okay, I have just a few words about our university. Maybe you don't know this area very much. It's our university was founded by Swedish king. Estonia was in those times was part of Swedish kingdom. We are very close to Sweden, so it's just this how do you call it, Baltic Sea. We are on the east side of Baltic Sea, Sweden is the west side, and Finland in the north side, and then it's Latvia, Lithuania, Germany, and Poland. So it's this Baltic area. And the university was founded in 1632, and it was actually the second university in the Swedish kingdom in those times, and it's one of the oldest universities in this area. And it flourished in 19th century because we had lots of professors of very tight connections with Germany, and we had very famous German professors worked in our university in those times. And of course, one of the most famous scientists who worked in our university and who also died in Estonia in our university was Karl von Baer, who actually discovered the oocytes of mammals. And he was born in Estonia, so he lived entire life in Estonia. And then we became occupied after the Second World War by Russians, and we were part of the Soviet Union, and of course all this stuff degenerated because we didn't have any investment for science, only for physics, because physics was meant for military purposes, so Soviet Union worked only for military purposes. Even for medical stuff, they didn't put very much investments because the purpose was only for military medicine or military aid. But we regained independence in early 90s, last century, and then after this regaining again, we already got some new horizons. We got EU membership in 2004, and we have Euro since 2013 or 2012. I think we have already for 2 or 3 years we already have Euro. And nowadays, I think we have quite modern infrastructure. We have got lots of investments. Even before EU membership, we got from World Bank and different kind of international organizations. And now we have got several investments from European Union. And medical faculty is one of the oldest, of course, as was part of university since the beginning. And currently we have university hospital is separate from university, I mean, as a legal entity. But there is very close connection because we are on the neighboring plots. And university hospital has 900 beds, 4,000 employees. University entirely has also 4,000 employees, so its university hospital is quite large, and it's regionally in southern part of Estonia is one of the most important hospital. Yeah, we even consider most important hospital in Estonia because there are several procedures that only can be done in university hospital. And about Estonia, capital is not Tartu. Tartu is university city in southern part, but capital is Tallinn. Tallinn is in northern part, and Tallinn is like 500,000 people. And Tallinn has, we joke it's like, I think it's like in Austria, capital has money, and small cities have brains and universities. So Tartu is also quite 100,000 people, and
the population of Tartu is 100,000.
100,000, yeah.
The population of Tallinn?
500.
500,000.
Yeah, 500,000. So it's What we do have, or I'm running in I'm running Department of Pathophysiology, and why I work in genomics is that as a part of this department we also have co-facility for genomic studies, and the equipment we have is 2 solid machines, basically 2 whole genome sequences, and we have done whole genome sequencing already several times, and we apply exome sequencing quite often, more often on continuously. The number of exome sequencing is increasing. We have it in the list of health insurance, so it's if you have like family or patient, you don't know what's wrong with him or her, and you want to find some unknown mutations, you can actually ask health insurance to pay for it. So they pay for it, and not very often, but they told that, let's say like 10-20 times a year would be fine, because it's quite expensive still. And maybe more interesting for medical studies that we have, for some disease collections, we already have quite nice disease banks of disease collections, and we continuously try to improve it. For instance, we have good collection for Parkinson's disease, for psoriasis, for vitiligo, also for osteoporosis, and some other larger diseases, but also for some rare diseases. For instance, osteogenesis imperfecta, what is very rare disease. We have collected all Estonian families, entire families. I mean, it's like trios, parents and children, and even many generations. So it's just to describe the infrastructure. And of course, one of the most famous infrastructure in Estonia is Estonian Genome Bank, where we have 50,000 DNAs and cells from volunteers, they are healthy volunteers, but we also, in addition to DNA, we collected some phenotype data. If they are smokers or non-smokers, smoking habits, drinking habits, also some else background, and all those data were confirmed by primary care doctors and family doctors. So it's basically it's not very heavily, very strong evidence, but there is still like documented diseases. And if you have more disease, you can go back. So it's like open database. So this is a building where I do work. It's a faculty of medicine. It's for preclinical departments. And this building was built like 15 years ago already. So it's you can see it's a four-story building, and this is just one building in the campus. What we have done with skin diseases. My background is I studied medicine, and when I got graduated, then I decided to PhD on neurosciences or in pharmacology, neuropharmacology. Then I switched to transgenic stuff. I have done or made several transgenic mice as a disease models, and I still have one PhD student who works on mice. But then I gradually switched to different diseases. So it's we work on Parkinson, and our group works, like half of my group works on skin diseases, and mostly psoriasis, because this collection is quite good, and we have quite many patients in Estonia, are easily accessible. We have been involved since 2003 with psoriasis, when I got my first PhD student in this, and there was just a young lady, and she asked how to genotype DNA, and she wants to do some genetics, and can I teach her? And then I thought, of course, it's very easy, but I didn't know very much about psoriasis at this time. So it's we have now collection of around 1,000 DNA, and we have quite easy access to patient pool in South Estonia. So it's because we are only for South Estonia almost only healthcare service provider. So it's around 300,000 people. Is a catchment area. But altogether in Estonia we have 45,000 psoriasis patients. So it's a very large number, and I just thought that this is good to collect at least like 5,000 or 10,000, because this is really what you need. You need like quite good. You need more patients for if you focused on DNA studies. We have done also some vitiligo studies, as my background is in neuroscience, and vitiligo is related to some neuroendocrine problems, because we don't know still why melanocytes die during vitiligo, and there is some hypothesis that melanocytes die because there is some connection with neuroendocrine processes, and we have done some works in this neuroendocrine profiling of vitiligo patients. But mostly we have studied psoriasis, because psoriasis is more straightforward, I would say. There is more information around, and it's more actively studied by other groups as well. And we are currently involved in 2 international consortia. One is in Europe and one is another in United States. And what I'd like to talk to you is about this system genetics approach. Well, our early studies, we were only focused on interleukin-10 family cytokines. And there is one subfamily in interleukin-10. Interleukin-10 is it's just another family of cytokines with quite precise and separate role compared to other interleukins, and there is quite clear indications that there is connection with psoriasis. The associations have been quite strong, were already sometimes quite strong by other groups. And then we found that recently was when we started to do in 2003 separate subfamily inside the interleukin-10 cytokines, what this was described. And this was interleukin-19 subfamily. And this subfamily contains 3 different cytokines: interleukin-19, interleukin-20, and interleukin-24. And you can see interleukin-10 is here, so they are all in one block of first chromosome, and they are all like good neighbors. But you can see that the structure is quite different. Interleukin-19 is the longest one. The area is the longest one, the genomic area where it's described, and it has different isoforms. We have found that there is maybe some impact of those isoforms. Why there are 2 types of isoforms? But today our early studies were focused only on those 3 cytokines. And just taking together all those studies, what we've done, we described 2 large haplotypes, or large haploblocks in human genome in those times. So it's basically the breakpoint for Caucasians, or somewhere here. It's in the distal part of IL-20 gene. And we discovered that some people who inherit this part, they have increased risk for psoriasis. And people who inherit some haplotypes from this part can actually have decreased risk for psoriasis. So basically we found in the genomes that in this area, depending that those 2 neighboring areas actually regulate or confer different kind of risks for psoriasis. Well, the risk numbers were not very large, but we were able to confirm this finding in different studies. Basically, the odds ratio what we found was 2.55 for this IL-19 and IL-20 haplotypes. This left side haplotype I showed, basically this haplotype, or this haploblock. And we found that while IL-19 and 20 are risk-bearing genes, then IL-24 turned out to be more protective. So its OR was quite small. Odds ratio was very small for certain haplotype, where it was IL-20 and 24 were combined. And as you see later, our more recent studies on RNA-seq also found some good relations with IL-19 and 20. So then genomics came in more and more. I started to do genomic studies in 2005. And at the beginning, we used gene chips, and I used lots of animal models because due to my background, and animals are very good to study because you can easily get tissues and you can very nicely design your study. With patients, it's more complicated because you really it takes ages to find perfect match always your group. So it's you have to take your sample as it is. But nowadays we use more RNA-seq, so we don't use gene chip very often anymore. And what's the major difference is that while gene chips are based on probes what are attached to solid support. Basically, it's like a glass or plastic support where you have lots of small oligonucleotides, and you hybridize your probe or your samples there. So it's basically the gene chip depends on prior knowledge. To make a chip, you have to know what genes you want to analyze. Even you know that in human genomes there are 24,000 genes. There are still very many new genes under the discovery, and or we don't know even that the genes do exist. And another thing is that very often we don't know the function of most of the genes. So I would say that from human genome, let's say less than 5% is well described. Most of it is very, very badly described, and we don't know what the genes are doing there. Especially we don't know about non-coding areas, and non-coding areas can't be accessed with gene chips. You can only analyze them with RNA-seq. RNA-seq is so considered as the most comprehensive technology. It used to be very expensive. Now it's getting cheaper and cheaper. Of course, it's not as cheap as gene chip yet. But what is very good is that you don't need to have a prior hypothesis. You just can ask a question like, why these patients are different from controls, for instance, or what is the difference if I look on the RNA, what's the difference between these patients and controls? So it's so-called hypothesis-free, and it's truly exploratory. Even you don't have hypothesis and idea what's behind it. After this study, you can actually generate one. You can really get an idea what's going on and where to go. And you don't need to do it. You can really take it like a preliminary study. You don't need to do it as already final study. Of course, you can design it so that you have 60 patients or your group is very large, but you have to consider that you lose lots of money with one go. It's always better to do like a small sample beforehand, and then do think, and then do next step. And good thing is that all RNAs can be detected in theory. It depends, of course, on certain technical nuisances, but basically, in theory, all RNAs can be detected. And it's why I like this RNA-seq is really with very large dynamic range. It means that with gene chips, you can lose genes with low expression profile. There is a certain detection limit. You just can't measure, or your error is already, detection error is so large that your results are not any so trustful. With RNA-seq, this difference between low and high expressing genes is very large. So this is a large dynamic range. In gene chips, it's quite narrow window. In RNA-seq, this dynamic range is very large. I think it's really basically there is no borders. How it works? We have RNA here, and then we have two ways. From RNA we can convert it to cDNA, or we can just fragment our RNA into fragments. And then when we have those small fragments, tiny fragments already, usually they are like 100 or 200 base pairs only, we attach blue and red adapters are. So we know this blue and red, the sequences are known, and then we just PCR amplify. And during this amplification, we can sequence them. And as we do it in parallel, in millions of reads, because all these RNAs, we get like not millions but billions of those reads, small fragments. It's really like a haystack, if you can imagine it. You take a blood sample, you extract your RNA, and then you have an unknown sample of RNA fragments. And then to get know what is inside, we just attach known sequences, and we start to sequence them. And during the sequencing, it's possible to separate them. We can say that this sequence comes from here, and we can really say, or we can identify the sequences. We can say that this is sequence number 1, number 2, number 3, and then we can list them. This is very important. This was very important technical solution, because otherwise you can mess up. You really don't know where those sequences are coming. And then what you need, if you get those very large files, these files can be really full one desktop computer very easily. If you get those files or those reads, then you need to have a reference genome. And for human, we have reference genome. For mouse, we have reference genome. And we get more and more reference genomes. So basically, they are your references where you compare your results. And comparing those results, you get those small reads, and then you can put together. You can say that these signals are coming from gene number 1 or gene number 2, and you can see it easily. Or normally, if you have like intron here, you won't get any match here. You get like half match here and half match here, and you can see that there is an intron. And this is also power of this RNA-seq technology, that it's like additional control. And very often you can see that there is sometimes introns are also read or used by some genes. And this is area where we will find new genes more and more. And at the end, what you get like this kind of picture where you have signal, and signal is RNA expression level is basically the read or the count number, how many numbers of RNAs you saw in your sample for certain genes. If you want to compare RNA-seq with microarrays, then you can see that microarrays have high run-to-run reproducibility, so they are very reliable, but the dynamic range is not very good. They are not able to detect alternative splicing. And what is also one power of RNA-seq technology is that you can find, or you can do de novo analysis of samples where you do not have reference genome. Basically, we have had one project where environmentalists or a guy from ecology department, they asked us, there is one bird, I even hadn't heard about this bird before, and there is no reference genome. And they asked how to analyze it. They want to do some monitoring, and so it's basically this RNA-seq helps to do it. So you can build up a new genome, and then you can use it for monitoring purposes or these kind of things. So psoriasis. We had 3 types of samples. We had psoriasis sample, lesional skin, and then from the same patient, we had non-lesional skin sample. And then we had another patient who was basically healthy control, not patient. And we built it up so that we had like triangles, 3 corners. This is lesional psoriasis, non-lesional psoriasis from the same patient. And then we had another patient who was control. And we built it up so that we had age matched and sex matched. We had 12 samples in each pool, 12 samples here, 12 here, and 12 here. This was design. And you can see after RNA-seq, we just looked if those samples are different in general. And you really can see that lesional samples are here, non-lesional are here, and control samples are here. And you can see that non-lesional are more similar to control samples. So they are closer. This group is closer than this, and this group is really more distant. And this is actually what you really expect, because lesional skin in psoriasis is very active and really very lesional, and very active inflammation is going on, while non-lesional, we consider non-lesional skin as healthy skin. Well, and first, I have a few tables, and first thing with this RNA-seq, what you can do, you can perform like statistical analysis. This was lesional skin compared to control skin. So basically, we extracted or subtracted control values of control patients or control persons from the values of lesional skin, and you can see here's the difference. And you can see if you can see minus sign here, then these genes are more active in controllers. But you see that most of the genes, this is just one fragment of this table. We saw 3,000 genes were differently regulated actually. So the p-value was very, very low. There is some problem with computer, but basically you can see that this p-value is very, very low. It's a 70. It's a 0.70 times 0, so it's 70 places after the comma, or 70 decimal points. So these differences are really huge, and just some genes. It's oligoadenylate synthetase, phospholipase, kali-granulated peptidase, And S100 is already well known. S100 peptides are very well known in psoriasis. Maybe these genes are not so well known. And what is recently came up was this late conified envelope gene. I think a few years ago, one Chinese group or group from Asia described CNVs in this area, and LC genes are getting more and more popular in psoriasis. But what we were more interested, because these kind of studies have been done also by other groups, and just a few months ago was recently published, I think by British scientists. And where we are more interested is actually this IL-1F9, this is interleukin 36 gamma. And you can see also the partners here, interleukin 36 receptor antagonist. These genes are really very heavily upregulated in this lesional skin. And this is something what it has been missed in previous papers entirely. And then we compared our samples. So I took those genes here. I took 50, because this table is only 25 or so, just for illustrative purposes. I took 50 genes from this table, and then I compared. I asked my computer to say me, what's the difference, how those genes differ in all those three groups, lesional skin, non-lesional skin, and controls, if I just look for these 50 genes. And you can see that all the 50 genes are different or basically upregulated. Dark spot means that it's upregulated. All they are upregulated in lesional skin. Here you can see lesional skin. While non-lesional controls, they are quite similar, and there is no difference, basically. Really, we can say that these 50 genes or these genes we saw in this table, they characterize lesional skin. Okay. Another question. Is there any difference between lesional and non-lesional skin? Should be, because they look different, and we consider that when we treat patients, then the lesional skin turns to be non-lesional skin, and we consider it's healthy. Well, you can see it's really, there is, again, huge difference, and you can see the same genes, but were also in this lesional compared to control. So basically, it seems that non-lesional skin really can be considered as a control skin. But you see that we don't see those interleukin. These genes are actually missing in this table. This means that these genes actually can differentiate controls from non-lesional skin. And when we used this comparison, then we decided to compare non-lesional skins with our control samples, just to see if those non-lesional skins are really real controls or not. And we found that we can find two types of genes. One genes are really like, here is lesional skin, non-lesional skin, and control skin. Some genes are really very similar to control samples. These genes, you can see that there is no difference. They are very similar. These red blocks and purple blocks are similar. And the only difference is between this green block here, what is lesional skin. But at the same time, some genes are more similar to lesional skin. Basically, it means that even if the skin is, if you look that the skin is clear and there is no lesion, there is a certain like background molecular pattern what describes this psoriasis. It's like a psoriasis susceptibility or how to say, preparedness for to get psoriatic lesion. That this skin is really vulnerable for certain physical and environmental challenges. And if it gets like some mechanical tearing or mechanical trauma, it can initiate psoriatic lesion. And what are those genes in this block? These are most interesting because they differentiate psoriatic skin from psoriatic patients from control patients. And you can see that the same genes like interleukin 1F6 is here, for instance, and interleukin 6 is here. So obviously there is some kind of some functional relatedness between those genes. And we for today we don't know what is the link behind those genes, because we just finished this analysis, so we haven't done more studies. Another thing what I forgot to show you is, if you look on those lesional skin sample, you can see IL-20 and 19 are very heavily upregulated also in those samples. So it again shows you the role of this 19 and 20, while 24 is not here. Okay, and as we were very much interested in this IL-36 family, there is another member is 37, and they all in one block. There is 9 or 10 genes, and they form one large block in first chromosome. And I plotted here 4 pictures: IL-F5, IL-1 F6, and IL-F9, what is 33G, and you can see that all those genes are very nicely, they correlate very nicely with the activity of process. This is a control, unlesional skin and lesional skin, and you can see, and you always see difference. Here is different, and here they are different. They are different, and they are different. Basically, in control skin, you don't see this gene at all. It's a 0 here. The same is here, it's a 0 or slightly above the 0. And here if you already have uninvolved skin, or just there is a patient without any lesion, with entirely normal skin, they already have elevated gene, elevated cytokine of this type of cytokine. While in control, just we took another cytokine from the same block, and you can see there is no correlation. This is just neighbor. It's IL-1 F7. It's inside the block. So this means that this correlation seems to be quite specific. And we only saw this correlation between those 3, while with this IL-1 F7, we didn't see this kind of correlation. You can see that here's a control, uninvolved skin and involved skin. It's even downregulated in involved skin. Okay, this is another large table. Just what we got very often is defensin beta 4A and carboxypeptidase A4. This is similar to what comes up with atopic dermatitis as well. So, and finally, what this genomic analysis actually helps us to do is we can really have a more systematic view. We can take all those genes we found differentially regulated, and we can say that, let's say our threshold is like on 2. We only look on the genes that are differentially regulated twofold. And then we can ask softwares, and we actually retrieve your databases, and we can ask, is there any correlative link between those genes? Or if you look on those genes, list of those genes, can you say what is the biological process behind this? And by using one of those different tools, there are tens of different analytical tools available. By using one of these tools, we found that this analytical tool tells that this is a psoriasis. It's very nicely correlates with psoriasis, psoriatic process. And you can see what green genes are downregulated and red genes are very upregulated. And why I like it's very busy picture, but it shows you actually the correlation or relation between different genes, where they are. Because here is extracellular space, it's a plasma membrane and cytoplasm. And if you want to find like extracellular markers for psoriasis, you have to look to this compartment, and you can see like this IL-36 is here, and IL-12 is actually downregulated. So it's quite convenient way to describe the process. To end the story with psoriasis, I think what is also a good idea, or what to do, is to look for patients or people, not even people to having comorbidities, but to analyze diseases who have some biological similarity. For instance, Crohn's disease with psoriasis, they have the same immune background. And we published recently one paper where we found similar genes that are affecting susceptibility for Crohn's disease and psoriatic patients. So we had population of for psoriasis patients, and then we collected Crohn's disease patients. This was like large international effort, and we described 11 different SNPs what actually correlated or indicated common risk for both disease. If you have this type of variation, you either can get you have increased risk to get Crohn's disease or psoriasis. And one quite recent paper we published in together with other with large international collaboration, that we found 50 new psoriasis susceptibility genes, while quite often or mostly those genes indicate the role of innate immunity. So this fits very well with RNA-seq data because this IL-1F family or interleukin-1F family is considered to be like innate immunity marker. Okay, now it's more recent than the study we did with Sandrine.
I'll give you a very short interruption. I
Yep.
think we have about 5 more minutes
Okay.
until we have to start with the clinical work.
Okay.
If you have a lot of more slides, let's just split them up and then talk about the rest
Yep.
in a smaller group, I think.
Yeah.
Just
Yeah.
to give you a little more.
Okay, yeah, thank you. I try to do it quickly. Just to show you, atopic dermatitis is considered quite similar to psoriasis. I don't know very much about atopic dermatitis, but I think there are some important differences still. And we ran this study so that we compared dermatitis samples and control samples, and you can see the list of the genes is quite different. You don't see very much similar genes here, and we still don't know why it's so. But we very often we see this gene here. I don't know if you have worked with atopic dermatitis more, maybe it's an important gene, and also carboxypeptidase A4. This was also similar to psoriasis, but for some reason we don't see very much interleukins here, but CD9, for instance, and some immune genes are here. And when we analyzed our data with this function network analysis, surprisingly, I got this machine told me that this is more or less similar to melanoma or how to say it, cancerogenic process. So it didn't recognize inflammation. It recognized inflammation, but for instance, in psoriasis it always shows you inflammation. But with atopic dermatitis, one hit was melanomagenesis or oncogenesis. So this was quite different compared to psoriasis. And as we had some small sample of ichthyosis vulgaris, we also were able to do ichthyosis comparison here. So you can see it's a conification. It's a late conified envelope gene, and it's clearly a conification problem. And we already see more immune-related genes. These are all 4. This block is related to immune function. Okay. So it's basically, this is my last slide. It's taken together also psoriasis studies show us that there is a clear link with innate immunity. And in case with atopic dermatitis, we have seen interleukin-17 pathway also activated, but for surprisingly, we got some melanoma activation or indication for melanoma. I don't know if AD is related to melanoma somehow. You can say it, because I don't have any idea. And I think that we need to do some further studies with atopic dermatitis. I consider this as a very preliminary result, but maybe we can discuss it later, what to do next. Thank you very much.